I'm Maria Wilson.
And I'm Danielle Mandikian.
And we are scientists. We love science.
Yeah, we do. So when we aren't doing it, the next best thing is to talk about science. And what's really awesome is that we're surrounded by some of the most brilliant minds in research.
We're going to step away from the labs today to talk to other scientists about the cool stuff they are thinking about, working on, and imagining.
As well as how some of these discoveries just might lead to new medicines. So grab your favorite drink, get ready to unlock your science brain, and join us for Two Scientists Walk Into a Bar.
The show for scientists, science geeks, and the people who love them.
What do you think the next breakthrough in oncology is going to be?
Oh, that's a great question. I don't know the answer to that.
Next step in oncology.
Gene cell therapy.
Yeah.
Or cell gene therapy, gonna work.
Yeah, I think cell and gene therapy.
I thought VEGF was gonna take care of everything, but it didn't.
Immunology.
I would be like checkpoints, but no, checkpoints were not a great success. I still feel it's checkpoints though. I love macrophages, so I have to say macrophages.
Yeah, so I'd say cell and gene therapy, so something personalized medicine, right? Something autologous, not even allogenic.
Hi everyone, welcome to the show. I'm Danielle Mandikian, and today we're going to be talking about the latest in the oncology space and what approaches scientists are actually taking to develop targeted cancer therapies. So I'm here with not one, but two experts in the field of oncology, one of which is a repeat guest, Fred de Sauvage. Welcome back, Fred.
Thank you.
Yeah. And we have first-time guest, Jennifer Schutzman, who's a medical director in early clinical development. Welcome to the show.
Thank you. It's nice to be here.
Thank you so much for joining us. So one of the ways that we've been starting off our conversations for this season is talking about unmet need in the space. Could we start off with y'all breaking down unmet need in cancer? You know, I think a lot of us don't really have the full picture because when we think about all of the research and resources that go into it, what are we still missing?
Yeah, I think it's true. There's been just tremendous advances in oncology. I think from a research perspective, all the way through how we conduct clinical trials and deliver medications to patients once they're approved. But I think, ultimately the goal in oncology is cure. It's to eradicate cancer. And I think despite the tremendous advances we've seen where we can extend life, where we have better therapies, more active therapies, new insights into how to combine these therapies, there still remains a need for more for patients who end up with very advanced disease, but also more importantly, therapies that we can really move earlier in the disease setting so that we have an opportunity to eradicate cancer.
Yep, for cancer research, it's been an incredibly exciting time. The progress have been huge in our understanding of the disease. We are able to target many of the driver of the disease, some of the main oncogenes, but there are still many others that remain to be targeted.
What are some of the new technologies that allow us to make progress? Like, why is it that now all of a sudden, we're dealing with these 3D structures and we're able to have that targeted molecule that can fit in the pocket? Is that driven by AI, helping to figure out more about structure, or how did we actually get to places like that?
All the above. I think in the case of KRAS, a famous scientist at UCSF had a very hypothesis-driven approach and really poke into the protein at the right place, that lysine 12 position that is often mutated to cause the aberrant activity of KRAS, and by literally poking on it, he found a pocket. Mm-hmm. But that's one way to do it. We've seen the Nobel Prize this year being awarded for the ability to predict the shape of protein. And now this is done at a very, very large scale. And we're going to have at our disposal for research the structure of pretty much all protein that exists. And I think more and more scientists will be able to predict where to fit a small molecule and design that with AI, probably come up with drug entirely designed by computational approaches.
We may not have been able to drug KRAS, say, specifically before, but researchers were able to identify molecules or agents to probe different proteins around KRAS to try to get at it, or different parts of how RAS may interact with other signaling molecules. And I think the PI3 kinase pathway is another great example where lots of learning over time has led to improvements in how molecules are designed, and even improvements upon molecules that have been successful at extending benefit to patients with those cancers.
Hi, Danielle.
Hey, Wellington.
Hey, Danielle.
What's up?
Definition. What is KRAS again?
It's basically a gene that encodes for this protein that's involved in cell signaling, and when it goes awry, cell signaling goes awry, and bada bing, bada boom, cancer.
Why is KRAS so hard to target?
That's a good question. I think you could look at it from two different perspectives. On one side, in the field, we target multiple proteins, and whatever success we have with targeting them, we think it's going to translate to other proteins, and that's just not the case. All of our proteins that we look after or we try to target are all unique, and that one was particularly hard because of the structure. So finding a point on it that we can actually target it and have access to it was kind of challenging.
And is it because the structure is this big spherical ball?
I don't know. Some spheres, if you need that outside, you're fine. But if it's like, kind of like closed in, it's those 3D structures that get us. That's why we need AI to help us with that. So, another thing that I really wanted to ask y'all about from both of your different perspectives in development is about new modalities. Everyone's just, I don't know what's the right phrase, like a buzz, and everyone's just excited.
Mm-hmm. Yeah, indeed. Part of the excitement is the many tools that we have at our disposal now to target these oncogenic drivers or many other aspects of a tumor cell's biology. So just to start, for example, from the RAS discussion that we just had, you can make a molecule that can target RAS that has many different properties. You can just block the activity of RAS by, again, fitting in that pocket that I've described to you. But in the process of doing that, you can also engineer additional property into that molecule. For example, one that people are pretty excited at this time is to engineer degradation. So that molecule is not only going to block the activity of RAS, but will target that protein, target RAS now to the machinery that is responsible for dismantling the protein, the proteasome. In the other aspect of targeting the tumor cells, again, the importance of combination is really to combine different approaches, not to combine multiple molecules on a target that are in the same signaling pathways that are being disrupted. I think probably that the more exciting approach is to combine molecule that will target different angles of the tumor. For example, recruiting the immune system to attack the tumor, disrupting the vasculature that's supposed to bring nutrient to the tumor. So many different aspects. I think the best result that we have is by combining these orthogonal modalities.
Yeah, I mean, I think there's a great example, right, from the PI3 kinase field where we understand that there's really connected signaling pathways that drive breast cancer, one being driven by mutated PI3 kinase, cyclin-dependent kinases, and then the estrogen receptor signaling pathway. And so combining the right therapies that deeply inhibit specific nodes in each of those three pathways can really have a tremendous benefit for patients.
So I think like a lot of people in general are kind of familiar with this idea of like tumor evasion or some kind of like maturation. So by combining these different strategies, does that help you kind of like chop off the different options a tumor would have to continue wreaking havoc?
I think it's one of the approaches that's exciting with new molecules entering the clinic, where, as Fred was outlining, right, we have new modalities, more mutant-selective molecules, things that are very specific for cancer and spare normal tissue. So with that greater safety, we have an opportunity now to combine different therapies and try to choke off different pathways that the cancer may try to get around if we just attack it on one angle. So I think that's one of the more exciting exciting approaches to the combination therapy, but really understanding what we're going after is critical. And that's why understanding the underlying biology, how that differs with different mutations that the cancer carries along with it, is really critical to inform the best strategy.
Sometimes I like to think about that also as kind of like the silver lining to the fails that we have in the lab, because you learn so much that you can then start to approach in a different way. And I feel like that's sometimes underappreciated.
Agree. And I think even clinically, right, we've had, again, progress made, but learnings along the way when that hasn't always been the progress that we've hoped for. And that has really, I think, informed the next steps for either that target or new combinations that we might want to bring to a specific disease, or even resistance that develops to therapies that we're trying in the clinic. That could be considered a failure, but it's also an opportunity for us to learn about how can we either address that resistance, or how can we even better prevent that resistance from ever developing.
It's worth pointing out that not only have the technologies to target the tumor cells, the pathways, the oncogenes have evolved, but the technologies that allow us to really understand the tumor cell, all those genomic approach, single cell approach and all that, allow us now to have a very, very deep understanding of a tumor. Literally, you can sequence every single cell that are present in the tumor, know about their DNA, their transcription profile, their epigenetic profile, their proteomic profile, even their location. We now have spatial transcriptomics, where you have section and you know where every single cell is located. Why is that cell located at the outer edge of the tumor? Why are these cells in the middle of the tumor? Why are those cells close to a blood vessel? The level of resolution and understanding now is, again, beyond what I had ever imagined 10 years ago. And we can do that on patient samples before we give them a drug to really understand and better try to select which drug we should treat them with, but also understand, the unfortunate, aspect of treating cancer is progression because of resistance, and then get biopsies from patients on progression and understand what has happened now. And literally, again, what we've seen there and how we understand those processes is just mind-boggling.
Yeah, it's really revolutionized our thinking. And I guess I would just add even more that we can even sometimes measure directly like what a drug is doing to a patient's tumor. And that can be really critical information to help us understand we're achieving what we anticipate or what we expect to achieve in the cancer, or we're not, and what else do we need to add to that drug to make it more effective. So I think there's lots of reasons that this really deep interrogation is critical to advancing some of the science and then also the clinical therapies.
Danielle, can you talk more about the idea of spatial transcriptomics and how that relates to helping us develop better treatment strategies?
Oh yeah, I mean, spatial anything is super interesting. First off, when you look at a tumor, it's not uniform. There's little pockets of things. So when you're looking at spatial transcriptomics, what you're looking at is what's the transcription profile of a cell in one area versus another area. When you think about that in terms of treatment, it helps you understand what the landscape you're actually trying to treat is. If you're trying to treat a gene that's only found in one part of the tumor, it's probably not going to be helpful. You want to look at something that might be more uniform, just as an example.
Is this another thing on the road to sort of a more personalized approach for medicines?
I think yes and no. I think at some level, there are types of therapies that might want to take in that approach to it, but it also helps us from a very basic research perspective. So Jennifer, from your perspective on the clinical side, where are we with using these new technologies in the clinic? How do we go from taking information from a patient biopsy and turning that into information that helps us inform a treatment?
Oh, I'm not sure if I can answer that, but I think it's not there quite today. In some aspects, I think it's moving in the right direction, right? There's blood-based testing, molecular testing that can be easily obtainable, and results can be available relatively quickly. So in normal clinical practice, those results could potentially inform the next treatment for a patient who's not responded to an initial effort at treatment. But I think there's much more. And what Fred is talking about in terms of this really deep understanding of what's going on, how cells are rewired, what is happening to the cell's neighbor that may allow it to grow, that we're not really quite ready for like implementation into like a clinical trial and ready sort of like just in time availability of data to act on. But I think it's something that we should be looking forward to in the future as these technologies improve, and our ability to understand how to implement those in the most impactful way improves as well.
And I also think it's a really cool perspective for people that are like earlier in their career and they're trying to think about where they want to specialize in. It's also a really neat concept to also be thinking about, like, I might be approaching something technical about, you know, whatever aspect of oncology that I'm trying to investigate, but also thinking about it from the end case. How could you make that something that's a translatable assay is a really important thing to think about early on. It's a very interesting kind of perspective to keep in mind.
Yeah, because it could really accelerate the learning, right, with that broader view. I think that's a great point.
Yeah. So one thing I also wanted to ask is, what do you think is one of the most interesting challenges for these newer modalities? Like, where are you really kind of passionate about seeing some improvement?
Yeah, I think that one aspect maybe that we haven't approached yet, that we very much would like to engineer in these new modalities or any molecule that we design today, is really the issue of safety and tolerability. Again, when we talk about developing resistance, it's something that gets worse and worse as you treat patients later in line. The key for prolonged response is really to try to treat those patients very early. And of course, that brings all the questions about detection. But if you're going to give one of these drugs early in line, you need very safe drugs. Patients will be on these drugs sometime for a long period of time. They're still very healthy. You want drugs that are very well tolerated. One way to achieve that is to design molecules that only affect a mutated oncoprotein, for example, that will leave the normal protein untouched, which is the protein that's present in the rest of your body, so it will only block it in your tumor. Of course, that's not always possible, and that's where you start to see side effect creeping up. But the best tolerated drugs are the ones that we can both use early in line and combine with other molecules, because these toxicities, of course, always add to each other.
Yeah, I would agree. I mean, I think we've seen a clear trend where introducing therapy as early in the disease process as possible has the biggest impact, but also means that patients can end up on these therapies for relatively long periods of time. And as Fred is talking about, certainly in a situation where there's very minimal disease and no symptoms from the disease, having extremely safe and well-tolerated medicines is critical. So I think there's ways that we need to continue to work on improving that and really test these new modalities to make sure that they're meeting that really high bar for safety, because our objective is to really push out these timelines, have patients on these highly effective, well-tolerated therapies that keep their disease controlled, or in the case that Fred's talking about, eradicate the disease.
So we have multiple, of course, other approaches now that you hear about that do that. Of course, you can do, as I said, mutant-specific protein, but delivery, for example, specifically to the tumor versus the normal tissue through antibody delivery approach. For example, you hear about antibody drug conjugates or T-cell bispecific antibodies. There's all kind of modalities now that people are working on to try to get the drug, if it's not fully specific in the first place for the tumor, trying to get it to the tumor through a specific warhead that's going to bring it specifically to the tumor cell.
That's my favorite place to be thinking about how to impact this type of development, because you have to have a system that's actually going to deliver it efficiently, and that means that it might not go just to the tumor. So having that driver to enrich it specifically there is really challenging.
Danielle, can you talk a little bit more about drug safety and tolerability issues as it relates to your work?
Yeah, I mean, not just my work. I guess I sit in a really small place where I'm looking at this, but overall, when we're trying to develop therapies, we want to balance out a dose that's going to be efficacious, but making sure we're well under the dose that could elicit some kind of toxicity. And so that kind of context of concentrations between good benefit, bad benefit, that's where we start to define what we call a therapeutic window. Like, what room do you actually have to do something well? Sometimes when you talk to people about oncology, you get like what we're having now, that spark of hope and excitement about everything that's coming out. And then sometimes you'll hit someone on a rough day and they're going to feel like, sometimes the oncology space can be dismal because we don't have as much success rate as we go to the clinic. What do you think from both of your different perspectives we can do to try to improve our success as we're moving therapies through development?
Give me a drink first.
Yeah. I don't know if you know this. You're actually going to be giving me career advice today, so help me out.
What can I say
That's
here?
a tough one. It is a great question.
My response to that is actually a little bit...
Now I got to hear it.
Yeah, the first thing to say is that, in oncology now, compared to pretty much every other disease, we have now an unbelievable number of models to study the disease, to study pretty much every cancer you can think about. They're genetically engineered models, patient-derived tumors that have been implanted. They're organoids that you've engineered or derived from patient's tumor that you can reimplant. So the number of possibilities here now is very, very large. There's two issues that we need to pay attention to when we test a drug in one of these models is really about how much activity do you really need to see to move forward in the clinic. And the second one is how well does the model recapitulate the patient population that you're going to treat. If you see activity in one model of lung cancer and then go and treat all comers in lung cancer, if that model had a very specific mutation that is present only in a couple of % of that lung cancer population, you're not going to pick up the signal. You need to enroll 100 patients to see 2 or 3 of these, and you will miss the activity. So you need to understand exactly why your molecule has activity in that model and select the right patient to treat with this drug. And it may sound easy like this, but in many cases, especially as you mentioned, if you're dealing with immune therapies, you may not have a situation as simple as a mutation. It's really about what is the immune environment of that tumor and so on, that really we struggle still today to define and to measure. And then, as I said, what's the level of activity? Is 20% tumor growth inhibition 100%, or do you need to see regression in your initial model to see a tumor shrinking in a patient? What's the level of activity that you need to translate? So still a very undefined or complicated question.
Yeah, I think from a clinical perspective, we try to design early clinical experiments based on what we know coming out of research. If the molecule is targeting a specific mutation, we try to identify a patient population where the cancer harbors that mutation. If there is a hypothesis around sort of co-occurring cancer phenotypes and inflamed phenotype, we will try to find that population. But some of the aspects of early development trials where we're initially testing the safety of very new molecules that have never entered humans before necessitate doing so in patients who may have already had the best available standard therapies before they go on to a trial like this. And so that naturally will introduce some heterogeneity into the patient population, into the cancer itself, and some of those pieces can also add to the difficulty on the clinical side to being able to interpret the signal. Like, what is enough to say we have an active molecule? Is a little bit of decreased growth of the tumor enough? Is a response based on clinical trial criteria, is that needed? Do we need a certain amount of follow-up to be able to really interpret the extent of benefit that we think this molecule could bring?
I want to make sure to also be able to have the opportunity to ask you, we got a chance to talk about where we are now, things that you're excited about. I'd also like to have an opportunity to ask you about where do you think we're going to be in 10, 15 years?
So first of all, if I look at myself and you would have asked me that question 10 or 15 years ago, I would never have been to predict where we are today. So it's always unexpected and much faster in terms of progress. So no matter what, it will be very exciting, and more exciting than I can probably even project it now. What I'd like to see is I still see all the pathways, targets that currently we are struggling to develop molecules against, and very much hope that we will have made great progress into that direction. The very specific delivery of drugs to the tumor, I think, would be a huge breakthrough, because we would be able to bring molecule there that also block the normal protein. In many cases, I mentioned it early on also, that many of the cancer have mutation, not gain of function mutation in an oncoprotein, but loss of function in a tumor suppressor. If that protein is not there anymore, you cannot target it. You need to target a dependency that's downstream of that one, but that will be a protein that's not mutated in your normal tissue, where it still plays a role. In that case, you will have the toxicity associated with targeting that protein. So can you deliver that protein specifically to the tumor? Again, I think there's huge progress that we can make there. Other modalities, delivery of antisense or other types of nucleic acid. There is many possibilities, I think, that I can see today, that I would greatly hope we will make great progress in the next decade. I think the more exciting part is those that I cannot see yet.
Yeah, I was also going to sort of point to tumor suppressors. I think that is still a very much unmet need or under-addressed area of oncology, of like how to deal with the loss of tumor suppressors. But I guess to me, maybe I'm a bit more modest in what I'm thinking, but I really think like all this deep learning that we are getting from advances in technology now, like bringing that into the clinic so that it is fully enabled in clinical practice and importantly in clinical trials, I see that as being a potentially really important advance to accelerate our ongoing development of new therapies. So I think that would bring us more robust information for faster decision making to really accelerate advances in oncology in the future.
Danielle, can you explain the role of oncoproteins versus tumor suppressors in cancer and the strategies scientists are using to target these issues?
Sure. I mean, look, when we're dealing with cancer, what we're dealing with is cell cycle. And for cells to go through the cycle of growth and division, there's proteins that are involved that start the process. And then there's proteins that are involved to kind of like, survey everything and make sure it's cool. So when in that context, when we're talking about oncoproteins, these are proteins where if they get messed up for whatever reason, it's going to result in that cell cycle check being lost and you get the progression to cancer cell. When we're talking about tumor suppressors, those are proteins where in their normal day-to-day function, they're putting on the brakes if something goes awry so that you don't develop cancer. Well, those can also get messed up. And once those are messed up, you lose the brakes. And so you're on that, again, back to that pathway of having uncontrolled cell division and therefore cancer. And so whenever we're thinking about this strategically, you have to figure out like what's going to be something that is, something you can target, that however it is that you're binding to it, you can elicit whatever you want to happen. So if it's a rogue protein that's really screwing things up, if we can try to remove that protein and get it cleared from the body, that becomes a great way to target an oncoprotein. Now for a system where it's a tumor suppressor where it got messed up, that's a lot more complicated on how you're actually going to achieve it. So I feel like easy explanation is get rid of the bad protein, but how do you fix a protein that was supposed to be there as your surveillance system? And that's where newer modalities and much more complicated treatment options start to come into play. What things do you think can improve the early clinical development experience from like maybe the patient side, or for what you would like to see happening for information for patients or their voice?
Yeah, no, I think that's a really important area for all of development, but particularly in early clinical development. I think we are paying more attention to the patient experience, the direct patient experience of these new medicines as they first enter humans. I think we've always collected what the doctor, the investigator, reports back to us as safety events, but we haven't always collected directly the patient's experience, and that information is really helpful for us to understand how tolerated these medicines are. Are they affecting a person's ability to complete their normal daily lives? Are they affecting their quality of life? We would be aiming for medicines that improve patients' quality of life. So hearing directly from the patient is really important from an early development perspective, and I'm really happy to see that incorporated even earlier in drug development now. I think the other place where there's been some real movement that's been positive from a patient perspective is inclusion of patient or patient groups in the considerations around eligibility criteria and study assessments. So what I mean by that, I can see the furrowed brow, is when we design a clinical trial, we often collect a lot of information, especially when a molecule has never been delivered to humans before. Now we seek input from patients, patient groups, to help us understand how our trial might be feasible from their perspective or not so feasible from their perspective, and what impact collecting all this information may have on them. It helps us understand not only the patient's perspective, but also to take another look and see what's really essential here, what's really important for our understanding of the drug. And it starts an early interaction with physicians, with patients, around what's important for them in terms of safety, clinical trial execution, and importantly, what they're looking for in new medicines.
Another kind of just emotional aspect that really strikes with me, having people in my family that have gone through this, is that also gives almost a sense of autonomy, because when you're going through it, it's just something totally out of your control. You have no say, you don't understand what your options are or whatever, so just being able to be present and be involved in it's got to be huge.
Yeah, I think again maybe a small sense of control or ownership, in contributing to the research that hopefully will impact an individual's lives, but also, individuals in the future.
Considering that the technologies that you need to use to diagnose, test tumors and all that are so complex now, so advanced, does it mean that patients further away from the major oncology center also benefit from those fancy approaches? Every tumor sequenced.
Yeah, I think there amazingly remains so much opportunity to improve on that, right? I think it takes a commitment on both sides, education and commitment for individuals, so that they know this is important, and it's not necessarily something that's out of their control or something scary, but something definitely in their control. And then a commitment on the side of medical centers, institutions, to be able to support that, to have the infrastructure, right, to provide the screening, to be able to provide the results and the appropriate counseling for patients. So there's actually a lot, I think, room for improvement, and a potential to have a much bigger impact.
For people that are listening, where can they look to be the most hopeful?
The fact that we have so many more opportunities to target the disease makes us hopeful that we will be able to affect the disease and to have progress for patients. Now, of course, as we've also discussed, there's different levels of opportunities. Obtaining full cure in the late line of fully metastatic cancer will be a lot more challenging. But the progress that we're making in early line, in early detection, or even in understanding, mind-boggling that we still don't fully know, understanding why do we get cancer, what do we need to change to not. The best way to cure cancer is not to get it,
Yeah.
To maybe even have opportunity to do preventive treatments that would eliminate tumor cells that are just appearing, before they develop. I think all these are realistic options that I think makes me very hopeful.
I'm hopeful. I feel inspired by this work. I have to say, on a big scale, what Fred is describing, I think there's lots of reasons to be hopeful when you look at oncology over the last decade, failures have led to successes. Advances have happened. Success hasn't been 100%, but I think, there's been persistence and courageous following of the science that's led to really tangible advances for patients. And I think we're on the right trajectory for the future. So I remain very optimistic, and I look forward to continuing to work with Fred on this mission.
Thank you both so much. This was a really interesting and just great opportunity to learn from you both. Thank you.
Thank you.
Thank you.
Danielle, I think this is one of my favorite conversations this season. Thank you so much. Looking ahead about technologies, modalities, and clinical considerations, what are you most excited about?
I'm really excited about the new modalities that are out there. And I'm talking about, where people are using kind of these Frankenstein type biologics or cell therapies. I think that these provide really interesting targeting and therapy strategies that we didn't necessarily have available to us in the past. And I think that it also provides really nice ways to control how we regulate whatever aberrations we're trying to deal with from a pathology perspective.
I really also enjoy when you talk to people who are on the clinical side or have clinical experience along with research. What's your feeling? So I guess I want to know what is your opinion about unmet needs in oncology, and what can we look forward to?
I think kind of whenever we have those conversations with both research and clinic, and we see how the research needs to be bridged to what's going to be practical in real world hospital settings, is critical. It kind of gives me the goosebumps, because I think that type of insight and kind of end-to-end planning definitely makes our likelihood of success much higher and more meaningful. And that's our show. Thanks so much for listening. If you haven't already, rate our podcast wherever you listen. It'll help new people find us. And make sure to subscribe. If you have any questions about the show, you can contact us at podcast@gene.com. And now for me, it's back to stalking cells.